Well, in this video, we will look at the Nobel Prize in Physiology of Medicine from the year 1904, awarded to Ivan Pavlov in recognition of his work on the physiology of digestion, through which the knowledge on vital aspects of the subject has been transformed and enlarged. Here you see the man at work with his assistants in his department, physiology department in St. Petersburg. The name of the lecture is Physiology of Digestion, and it is summarized by me here. It's quite a long lecture, and even the summary is quite long. But still, if you want a complete lecture, then you go, of course, to the official Nobel Prize site. Here we go. The food we eat decomposes and enters into combination and dissociates again. This is the subject of physiology. The first stage foods must pass the digestive canal to be digested. The digestive canal is in fact the external surface of the body turned inwards, thus hidden in the organism. It consists of a number of chemical laboratories. The muscle wall of the digestive channel forms a mechanical apparatus, and it takes care of the passage, detaining and expelling of the food and mixing it. Glandular tissue is also part of this wall or lies beyond it in glands, putting via tubes into digestive tract. Juices into the digestive tract. Some of these substances, ferments, are found only in living organisms and break up the food with such ease as no other chemically substance could do. In order to investigate the product of secretion in the big digestive glands, we cut from the wall of the digestive canal small pieces, in the center of which were the normal openings of the secretory ducts. We then closed the opening in the wall of the canal by stitching, and the excised pieces were sutured to the corresponding site on the surface of the skin with the openings of the secretory ducts to the outside. The juice was diverted from the digestive canal and could be collected in special vessels. In order to collect the juice produced by those microscopic glands that are located directly in the wall of the digestive canal, already since a long time large pieces were cut out from the wall of the digestive canal, and artificial pouches with openings to the outside were made. The defect in the digestive canal, of course, was closed by stitching. The digestive canal is a series of separate chemical laboratories. I cut the connections between them in order to investigate the cause of phenomena in each particular laboratory. Thus I resolved the digestive canal into several separate parts. This, of course, necessitated laying short and convenient passageways from the outside into each separate laboratory. For this purpose, metal tubes have long been in use. These coalesce with the skin in artificial body openings, and during the intervals between the experiments they can be sealed by means of plugs. We did our best to harm animals as little as possible and keep them happy. Pavlov shows how, but it's skipped here. It's already going to be long. But it's interesting. You can read it on the official site. Observing dogs, we noting the kind of substances getting into the digestive canal as well as its competition determined the work of the digestive glands, the amount of reagents produced, the condition. Pavlov gives examples. I also skipped here. Again, interesting, but for time reasons. Edible substances evoke the secretion of thick concentrated saliva. Why? It enables the food to pass through the digestive tube. Certain disagreeable substances made the glands secrete fluid saliva, apparently either to dilute these substances and to cleanse the mouth from such substances. Only water is secreted. Dry bread evokes secretion of more saliva than meat. Eating dry bread requires saliva to dissolve the components of the bread and to recognize its taste, to soften the hard and the dry bread. Inside the stomach, the same bread proteins induces secretion of more protein ferments than the proteins of milk or meat. Here again a look at the laboratory in St. Petersburg. There are many examples of similar purposeful relationships between the work of the digestive glands and the properties of the substance entering the digestive tract. It is clear to all that animal organism is a highly complex system consisting of an almost infinite series of paths connected both with one another and as a total complex with the surrounding world with which it is in a state of equilibrium, a condition for existence. How is this equilibrium affected? Apparently, it should be assumed that in some way certain properties of the object act on the gland, evoke in it a specific reaction, and cause its specific activity. Analysis of this influence on the gland is an extremely intricate matter and one requires much time. By means of a metal tube, we introduce meat into the empty and inactive stomach of a dog without the animal noticing it. In a few minutes, the gastric reagent, an acid solution of the gastric protein ferment, begins to exude from the walls of the stomach. Is this action caused mechanically? No. It is the components of meat soluble in water that are the stimulating substances, but much we don't know. A few minutes after the thyme finds its way into the next section of the digestive channel, into the duodenum, one of the glands of this section comes into action. This is the pancreas, a large organ located at the side of the digestive tract and connected with it by an excretory duct. But which of the properties of the thyme advancing in the intestine acts as a stimulating agent? Not by the juice which joined it in the stomach, but by its acid content. The reason? The gastric laboratory uses its protein ferment under an acid reaction, but pancreatic ferments cannot develop their activity in an acid medium. The pancreatic juice acts above all as a solution of soda to create a neutral environment. This should be E. It has been known for a long time that the pancreatic juice contains all the three, a protein, a starch, and a fat ferment. The protein ferment in the pancreatic juice is at least sometimes an inactive latent form because the protein ferment destroyed them. We established that the walls of the upper section of the intestine secretes a special fermentive substance, the action of which is to transform the inactive pancreatic protein ferment into an active one. The acting ferment, upon coming into contact with the protein substance of the food in the intestine, loses its half-moon action with regard to the other ferments. How does the given property of the object, the given stimulant, reach the glandular tissue itself? The system of the organism is united into a single entity in two ways, by means of a specific tissue which exists solely for the purpose of maintaining interrelations, and by means of the body fluids bathing in all body elements. Long before us, it was established that the work of the salivary glands is regulated by a complex nervous apparatus. This is designated as a reflex or a reflex stimulation. Normally, this reflex is always of a specific nature, that is, the endings of a centripetal nerves receiving the stimulation are different, each bring about a reflex only when there are very defined external stimuli. Accordingly, the stimuli reaching the glandular cell must also be a special peculiar one. The discovery of the nervous apparatus of the salivary glands immediately impelled physiologists to seek a similar apparatus in other glands lying deeper in the digestive canal. Despite great efforts, no positive results could be achieved in this respect for a long time. Physiology has, at last, gained control over the nerves which stimulate the gastric glands and the pancreas. We stimulated the nerves of animals that easily stood on their own feet. Our experiments not only proved the existence of a nervous apparatus in the above-mentioned glands, but also disclosed some facts clearly showing the participation of these nerves in normal activity. An example, when the connection mouth-stomach is severed, no food can pass through. When the dog gets food in its mouth, the stomach reacts and secretes juice. The dogs can still be given food directly into the stomach. The gastric juice serves the purpose of the gastric glands research. Here we, by the way, see the Nobel Prize diploma of Mr. Pavlov. Okay, that juice is also a good remedy in treating patients suffering from insufficiency. It is clear that the mere act of eating, the food not reaching the stomach, determines the stimulation of the gastric glands, no longer if the so-called vagus nervus is severed in the neck of the dogs. Of course, special measures were taken to avoid harm for the dogs resulting from the severing. One kind of stimulators of the digestive has not yet been mentioned. It has long been known for sure that the sight of tasty food makes a hungry man's mouth water. Also, lack of appetite has always been regarded as an undesirable phenomenon. In physiology, too, psychical stimulation of both the salivary and the gastric glands has been reported, but far from being generally admitted. Appetite, craving for food, is a constant and powerful stimulator of the gastric glands. At the mere sight of food, nervous and excitable animals secrete several hundred sedate and quiet animals only a few cubic centimeters of gastric juice. An extract and frequently repeated analysis of this, the above-mentioned sham food experiment, convinces us that in this case the secretion of juice cannot be regarded as being the result of a simple reflex stimulation of the mouth and throat by the food swallowed. Is the oral surface stimulated not by arbitrary chemical substances but only by specific ones contained in the food consumed? The answer is no. Each kind of food ingested by the dogs during the experiment acts as a strong stimulus only when it suits the dog's taste. When we introduce bread into a dog's stomach through the metal tube so as to prevent the dog from noticing it, the stomach does not react in any way for plus minus an hour. For example, the sight of dry bread evokes a stronger secretion of saliva than the sight of meat, although the latter, judging from the animal's movement, may arouse considerably keener interest. In short, experiments with psychical stimulation represent an exact but reduced copy of those with physiological stimulation of the glands using the same substances. With regard to the action of the salivary glands, psychology has taken its place besides physiology. At first sight, the psychological aspects of this action seems to be even more indisputable than the physiological. Appetite not only generally acts as a stimulus on these glands, but also that it excites them in a varying degree according to its direction. For the salivary glands, the rule holds that all variations in their activity observed in the physiological experiments are exactly duplicated in experiments using psychical stimulation. Pavlov gives more examples, which for time reasons I've skipped it, but they're interesting. You want to know them, read the original lecture. What now is the physiologist to do with the psychical phenomena? Disregarding them is impossible, since in the action of the salivary gland, which we are interested, they are closely connected with the purely physiological phenomena. If nevertheless the physiologist wants to study them, he finds himself faced with the question, how? We wanted to remain physiologists instead of becoming psychologists, and decided to take an entirely objective point of view. Again, he gives examples which for time reasons are skipped here. Again, you see the man at work in his laboratory. Then we pass on to little theoretical considerations. The given fact fit readily in our physiological way of thinking. Our stimuli working from a distance may rightly be termed and regarded as reflexes. It appears that the activity of the salivary gland is always excited by some external phenomenon, that it is induced by external stimuli like the usual physiological salivary reflex. Only the second is evoked from the oral surface. The first, however, from the eye or from the nose of the dog, etc. The difference between the two reflexes is firstly that our old physiological reflex is constant, unconditioned, while the new reflex continually fluctuates and hence is conditioned. Essential differences between the two reflexes can be observed. In the unconditioned reflex, those properties of the object act as stimuli with which the saliva is confronted physiologically, for instance, hardness, dryness, certain chemical properties. In the conditioned reflex, however, those properties of the object act as stimuli that in themselves have no direct relation at all with the physiological role of the saliva, for instance, color. These last properties appear here in some way as signals for the first. One is bound to regard their stimulating action as a further more delicate adaptation of the salivary glands to the phenomena in the external world. Here's an example. We intend to pour acid into the dog's mouth. To protect the oral mucous membrane, it is apparently very desirable that saliva accumulates before the acid enters the mouth. On the one hand, the saliva prevents direct contact of the acid with the mucous membrane, and on the other hand, it immediately dilutes the acid, thus generally weakening its inverse chemical action. However, essentially, the signals have only a conditioned significance. They change readily, and also the signalized objects cannot come into contact with the oral mucous membrane. Consequently, the final adaptations would have to consist in the fact that the properties of the object serving as a signal sometimes do and at other times do not stimulate the salivary glands. Any phenomenon in the external world can be made a temporary signal of the object stimulating the salivary glands, if the stimulation of the oral mucous membrane by this object has been associated repeatedly once or several times with the action of given external phenomenon or on other sensitive body surfaces. On the other hand, promptly active signals can lose their stimulating effect if repeated over a long time without bringing the object concerned into contact with the oral mucous membrane. When the most usual kinds of food are shown to the dog for days and weeks without letting me eat, the sight of the food will finally cease to evoke salivary secretion. The mechanism of stimulating the salivary glands through the signal characteristics of the objects, the mechanism of the conditioned stimulation, can be easily conceived from a physiological point of view as a function of the nervous system. As we have seen, each conditioned reflex, each simulation through the signaling characteristics of the object is based on an unconditioned reflex. That is a stimulation through the essential characteristics of the object. So it must be assumed that the point of the central nervous system that is being stimulated strongly during unconditioned reflex diverts itself the weaker stimuli exerted by the external world on all points of the central nervous system. That is due to the unconditioned reflex, a temporarily incidental path is created for all other external stimuli towards the central point of this reflex. The conditions influencing the opening and closing of this path is being in usual tissues constitute the internal mechanism of the signaling characteristics of external objects being effective or ineffective. The physiological basis of the finest reactivity of the living matter, the most delicate adaptability of the animal organism. Okay, this was it. As said, this is a summary. The original lecture is quite long. If you want to nevertheless completely read it, then you follow this link, which is also in the description of the video. Yeah. Okay. Thank you for watching. Here again, you see a Pavlovian experiment with a dog, as described. There's also another video on this Nobel Prize with more general information. And next week we'll move on to the year 1905 for medicine, physiology, but also for chemistry and physics.